all managed to see some of the poster presentations that were going on over lunch, which is, well, was it lunch, which is great anyway. I guess it's somewhat stolen the thunder, but I guess the interesting bit in this presentation will be to give the overview about where everything fits together. And our program is really around looking at the role of powder agglomeration, and particularly we're interested in microparticles and the behaviour of microparticles that pose some big challenges. As you've already seen, there's a variety of partners involved in our program in Form 2020. The University of Hertfordshire, but also with our colleagues up in Leeds who look at the molecular and crystal modelling, our colleagues at Cambridge that are looking at powder modelling and powder flow modelling, as well as the experimental techniques. We worked with people at Bath around particle characterisation, single particle characterisation techniques. They've since left the consortium and moved into private industry, in fact. And then our colleagues at Manchester have been looking around XRM, and we work with a variety of companies that cover both generic formulation development companies such as 3M, but also innovator companies like AstraZeneca and GSK, and a variety of instrument development technologists like Malvern Analytical and Zeiss microscopy. So we've worked with people across the pharmaceutical supply chain, including overseas generics, overseas materials, synthetic industries, as well as the end user industry. So we try to work all the way along the pipeline of inhaled drug delivery, and that's why we really needed such a big cohort. I think it's been shown some of the challenges that come with large multi-partner collaborations, some partners pulling out, and now we've ended up in the middle of a pandemic just at the point when we were about to make our killer experiments, but that at least passes some nice findings off till after we're all back in the labs. If we look at really the challenge around inhaled formulations, we can see some of these typical formulations here in this slide. So in some cases, we've got these agglomerated granules that are formed from micronized particles, or we have these larger diluent particles that contain the micronized drug on their surfaces. But really the aim of all of this is to be able to densify down an aerosol. So an aerosol is a dispersion of colloidal particles in air, but to be a medicine, we need to densify it down into a typical pressurized metered dose inhaler or a dry powder inhaler. And the real key challenge for it is how we can condense that phase down reproducibly. So we get a reproducible fill weights into our devices, but also then regenerate this aerosol cloud on demand. So where we set out on this program was to see, could we develop predictive models of formulation behavior, the manufacturing processes that are needed to make those formulations, but then also the aerosolization events. And really it's about increasing the understanding of product performance and stability, because if we can achieve that, then it should be easier for us to accelerate new products through development, but also to understand the failure modes of those products. And if we think about the overall aim, it's about trying to get the drug into the lung, into the lungs to treat these patients. And if we think about the performance, typically, a lot of the drug actually ends up here in the throat, the mouth throat of the patient, rather than getting out into these regions of the lung, which are of most interest to us. But the real issue for us is about the variability in this performance. It's quite variable from patient to patient, and it's quite variable from dose to dose. And it's about trying to understand the links between that and how we can better engineer those formulations. So we start with a pre-aerosolized formulation, typically the powder in a blister or a capsule. And it's only by combining that with the device and the device mechanics that affect the airflow and the patient's lung function that generates that aerosol. And we're typically able to characterize that post-aerosolized formulation and relate the properties of this aerosol itself into the lungs. But the links between what's stuck inside the device and the development of the aerosols going to the lungs is less well understood. And most of the work, it's true to say, focuses on characterizing the properties of the aerosol. If we understand the properties of the aerosol and its microstructure, then we can understand what's going to happen when that aerosol gets into the lungs. And there's been some really nice work from our previous partners down at Bath, Robert Price, looking at characterizing the microstructure of these post-aerosolized formulations. But there's still this black hole, if you like, about the structure in the pre-aerosolized formulation. How does this structure here influence the post-aerosolized formulation? And therefore, how does it influence what gets into the patient's lungs? And that's really the area where we focused on in this project in the end, is around these characterizing these pre-aerosolized formulations and trying to link that to the performance. And if we think about these formulations and we try to apply a material science predictive approach to it, really we're trying to understand this final vertex of the material science tetrahedron. We're able to measure the properties of the individual components very well. We're able to measure the performance very well, and we're able to control the process. So the bit that's really missing is understanding the structure of that formulation. And even with good patient technique, the influence of the powder microstructure is massive on performance. And this is the one area that we've really struggled to predict, the area that we struggle to understand, and the area that we struggle to measure for really 20 to 30 years in dry powder inhalers. And this is the big barrier to trying to understand the reproducibility of performance. So if we think then and look at the inhalation behaviour of powders, well, this is a slide that you've seen somewhat earlier. Early on in this programme, we did some computational modelling of the emission of aerosols. So it was a CFD-based approach to optimise inhalers. And to validate that work, we undertook these experimental studies of studying fluidization. So we built these fluidization rigs, and these rigs are to scale of what's inside a typical DPI. So the air can be drawn through this inlet. It aerosolizes the powder that's been plugged inside this blister simulant, and then is emitted. And we study the fluidization and the entrainment of the powder out of that device. And we can pull respiratory profiles through. So you can see here by a coordinated pulling of this piston, we're able to draw an airflow, and it's a simulated human airflow through this device, and study the entrainment behavior. And the whole time we study this using high-speed videography, and we're able to build up a model of the powder emission. And when we do that, we saw some pretty good fits with the bulk diluent lactose. But when we started to bring in materials which were much more representative of inhalation formulations, where there's a component of fine particles. In this case, it's a material called Lactohale 200 that has roughly about 20% of particles below about 30 microns or so. We can see that there's a quite chaotic behavior in the aerosolization events. So you can see throughout the video, you see plugs of powder that become emitted. In some instances, and in other powder fills, the powder doesn't even empty at all in the same time frame. So it's a very variable process. So it's a highly chaotic, a highly variable fluidization process for those lactose mixtures that contain those fine micronized particles. So really we wanted to start looking to see what governs the powder cohesion. And some of the work which Zhizhong presented on his poster shows how we've improved these initial CFD models. So by developing up his DEM simulations and being able to undertake a design experimentation approach with the simulation, we can start to look at things like changing the Hammaker constant, changing the particle shape and particle asperities, to see whether it's whether the DEM approach is able to simulate these high fine content powders. And you can see from the simulation results on the right that yes, we are able to simulate effectively the chaotic and the fragmentation approach of some of these cohesive powder mixtures. But we're now also able to start doing things like building in different powder cohesion forces. So the real trick is whether it's going to be possible to include accurate powder structures rather than simulated powder structures, and cohesive force balances. So if we can take our laboratory measurements of surface energy forces, can we build that into the powder cohesion model to try and model better the fluidization and aerosolization events? So if we're able to model this, how can we go about understanding the structure of these powders? What approaches have we been able to bring to light around measuring powder microstructure? Because it's that powder microstructure that we need to build into those powder flow and CFD models. And where we started with this was with trying to deconstruct the powder microstructure using X-ray tomographic approaches. So starting with the X-ray source, sticking a representative sample, for example, powder in a capsule, into our X-ray microscope, building up those radiograph images in multiple, by rotating the capsule, we can build up a three-dimensional image of that powder, and then we reconstruct this model of a powder. We can zoom in on that, we can look at it from different scales of scrutiny depending on the instrument which we use. And as Parmesh pointed out in this poster, this has recently been published, our first journal publications from the programme. And you can see the types of information that we can get from this XRCT that we've never seen before from any of the previous types of approaches, whether it's optical microscopy or scanning electron microscopy. We can start to see the spatial arrangements of the particles within these powder blends. And this is, typical of the arrangement of powder within a capsule. We can see where the powder is located, we can see the density, we can see the porosity of this powder bed, and we can also start to pull out statistics like particle size distribution. So this is really the first time that we've been able to look at materials on the scale of inhalation formulations using three-dimensional imaging techniques, which is really quite exciting for us. We can start to pull out, as Parmesh said, statistics of particle size distribution, but we can also now start to separate the impact of preferred orientation. So many of the particle size techniques that have been available to date show a preferred orientation of the particles as they move through lasers or as they settle onto microscope plates. And the XCT approach images that powder as it's present in the bulk material, and we're able to pull out particle size distributions of that powder as it appears within the capsule. And this removes a lot of the artifact of the typical particle size distribution techniques. So that's the major component of the formulations, which is the carrier or the diluent lactose materials. But we also then, within our powders, have agglomerated material, and there are multiple formulation components. So if we think about our typical formulation components, and really up until our program, this was the only way in which we were able to image the materials that were present and pull out information on their shapes and connectivity was in 2 dimensions using scanning electron microscopy. So we have this large particle size carrier or diluent lactose. We'll typically have a micronized API. So this shows a number of larger agglomerates of very small individual drug particles. Those particles are between about 0.5 to 5 microns each. But we may also have extra micronized lactose within that formulation as a formulation aid. So up till now, it's been really difficult to see what this microstructure really is. And certainly it's been impossible to see the microstructure of undisturbed agglomerates, those agglomerates which mix with the lactose, which are mixed with the lactose and go into the formulation. So this shows the imaging that we've done with nano-CT and published recently at the RDD conference in the States with our colleagues from Zeiss. We can start to see the particle distribution within a single agglomerate of micronized particles. And you can see the scale here. Some of these individual particles are on the scale of about 200 to 300 nanometers. And we're able to now start to see the connectivity, the particle size distribution within those agglomerates, and start to build up an image of what the forces holding that agglomerate together are. And when we do this, we're able to pull out a particle size distribution, which we see on the left here. And we can see that there are particles which are down as low as 200 nanometers, but the bulk of the particles are within the range of about 2 to 3 microns. So previously, if we'd sized this, we would have seen particles with a 2 to 3 micron size, but because of the XRM, we're able to now look at the individual agglomerates and see that they're roughly 70 to 100 microns in the length dimension, but that's composed of a high number of individual particles. We can also start to calculate out the porosity of this, and we can see that there's a porosity of about 71%. So really, there's only almost 30% of this agglomerate which consists of the particles, but yet it's very resistant to being broken up in the airflow and during the blending processes. So if we take then those agglomerates, how do we formulate those? Well, we'll typically take an excipient, this lactose monohydrate diluent. We'll take a micronized API, an active pharmaceutical ingredient, and that's in the concentration range of about 0 to 20%. And nowadays, it's most common to undertake a high shear mixing approach. So we took a variety of these blends and an experimental design. We're just going to show you one of these throughout the results. We used the PicoMix, which is a small scale simulant of an industrial scale high shear mixer. Subjected it to 1,000 RPM mixing high shear for 2 minutes, which we've taken as a standard from previous literature studies using the PicoMix with good effects. And when we do that and we start to image these powders, I'm seeing there's some questions coming in here, which I will pick up as we go through towards the end of the presentation. When we image this substance in 3D, these blends in 3D, we can start to pick out individual lactose crystals. They're those large particles, but we can also start to see these diffuse regions around the particle coating. So because of the density difference of these porous areas, we're able to pick out what is the drug coating phase around the coarse lactose particles. And when we zoom in on those and we look at this particle from two orientations, you can see this buildup of a thick layer of micronized drug around those coating particles. And when we look at this from a different dimension, so it's the same particle to a slightly different slice, we can see that there's a really face-specific accumulation of the drug on these lactose carrier particles following the blending. So our X-ray microscopy has been able to reveal where it is that the drug is distributed throughout the blend, but it's also able to look at microstructural effects, such as the interaction of drug on the surface. And when we can digitally remove the rest of the formulation from the image, and we can look at a single tomahawk, these typical tomahawk shaped crystals of lactose monohydrate. And on these particles, we're able to see the face-specific binding now of the drug phase much more clearly. So you can see there's a very thick layer of drug building up on this particular face of the lactose crystal. And we can see it building up on the opposite. We can also see what we think is a small lactose crystal here, but of course, if we turn that into another dimension, we'll be able to characterize that much more clearly. So we see less API on some facets. We see significant amounts of API on other facets, such as here. And that's now starting to help us build up an understanding of which particle-particle contacts are going to be important in terms of binding drug within the formulations. And we'll be able to start to carry out modeling, such as how changing the shape of that lactose crystal through controlled crystallization would alter the formulation performance and formulation blending behavior. So the next question then that we ask ourselves is, having seen that the drug does bind to specific particle surfaces in our diluent, and this is a two-component formulation, it's simply the large carrier lactose and small micronized particles of the API, we'd be interested to see whether we can work out why it is that it's sticking and adhering to individual drug crystals, our drug crystal faces. So work which we're just about to publish, but we've published some of the preliminary findings again at the RDD conference just a month ago, is to carry out laboratory diffraction contrast tomography approach. So we can see some of this first work that we've undertaken here. We've got a Kapton tube that contains crystallized hexamine, which our colleagues up at Leeds very carefully controlled the crystallization approach. We can see just a two-dimensional slice through one particular plane of that sample here, showing the individual crystals of hexamine. But at the same time, we build up a diffraction image at a particular orientation of the powder, and we build up that diffraction image, which is the powder diffraction spots. So it's possible using the patented algorithms from Exnova that work with the Zeiss instruments, and I believe these are the only instruments that are capable of carrying out this analysis. We can carry out back calculation algorithms to show what the orientation of a crystal is to produce these diffraction spots. So we're able to now identify the specific crystal within this powder blend from the diffraction spots, and we're able to identify what the crystal plane orientations are from those diffraction spots. And when we do that, we're able to identify individual particles within this powder blend that differ only in their orientation within the powder. So we can see here with this sample of the hexamine, for example, we can see there's a variety of different orientations of these particles which can be identified. And hexamine was a relatively good example to go for, because if we pull out these individual crystals of hexamine, there's only really one family of faces on the hexamine crystal. So it's a reasonable test bed for us to start with. But what we can see is that these particles, the 101 family of crystalline faces, are oriented in a variety of different orientations, and that's how they're colored here. So in this centralized image here in the center of the screen. So if we're able to identify the orientation of the particles, And we're able to identify the individual crystal faces or face families of those particles. And we're now able to superimpose on top of that the micronized particle phase that we pick out from our phase contrast. We'll be able to identify which particular crystal faces or family of crystal faces the majority of the drug adheres to. The first step on this work, and this was some of the work that we were doing before everything got shut down for the viral shutdown, is an image of a single lactose crystal that's been mounted on a nice toothpick here, and we've carried out the diffraction analysis, the DCT experiment on this single crystal of lactose. We can see here the simulated diffraction spots for that particular crystal, and from that we're able to work out the orientation. So you can see we're somewhere along this step of being able to pick out the orientation of a particle based on its crystallographic diffraction results. So the next stage for us is to move this on and start to look at multiple particles at the same time, and to try and see whether we can index the crystalline faces to which the drug adheres. So having identified ways of looking at the powder, having identified ways of characterizing agglomerate behavior and looking at agglomerate thicknesses within the blends, and to be able to start identifying where those drugs stick, one stream of this project is to look and see whether we can identify cohesive and adhesive force balances within these formulations. And this is some of the work which has been carried on up in Leeds in combination with my colleagues at Hertfordshire as well. So the first step really was around simulating the crystal behavior of lactose monohydrate. And you can see from this work, which we've again published in Digital RDD, being able to index individual crystal faces and characterize the surface energy of those individual crystal faces. So from this, we're able to identify what the surface energies are between individual crystal faces, for example, and we were able to calculate out adhesion force measurements, similar to as we published previously in 2014 in this work before the project started. We're able to work out attachment forces, dispersive surface energies, and total surface energies across the multiple faces of the surface. So that means we can now work out the binding forces or the adhesion forces of our drug onto each individual crystal face. And if we know which crystal faces are presenting in our powder blend, then we're able to understand the detachment force that's going to be necessary during aerosolization. Vivian presented just earlier on in the poster session his modelling on fluticasone propionate, which has been used to predict the crystal interfaces of the drug crystal, this fluticasone propionate drug crystal, and also to work out the surface energy contributions from that. We've also done the work, and I'm zooming over this obviously, which kind of doesn't do any justice to the volume of work which the scientists and the PDRAs have done. He undertook a crystallization experiment and analysis on terbutylene sulfate. This is the second drug which we use in our systems, and has been able to calculate out the surface energies, dispersive surface energies, and surface energy distributions. And I guess the really interesting thing for us from this work was that it was obvious that our experimental techniques weren't able to probe the high surface area regions of the crystals of drug. It was really only picking out, even at pretty high surface coverages of 12% or so, it really wasn't picking out, it was really only picking out the very high surface energy regions. And that's quite important to us, because the high surface energy regions are actually a very small proportion of the drug crystals which are being formulated. So it wasn't really giving us a very good understanding of how these materials were going to behave. So again, that's work which we've now just had published in CrystEng.com as our first publication from this project on the crystal modelling work. So if we now start to reconstruct those powder blends, we can identify the individual large component of the products, the lactose monohydrate. We can identify how those drugs agglomerate. We can characterize the distributions of particle sizes within there. We can characterize where the drug distributes within the powder blends. I guess it's about reconstructing those powder agglomerates within a blend. So this shows an example of correlative microscopy. I think we've looked at this video in previous work, but what we're doing here is identifying where the fine particles are distributed throughout that blend. So we can see this is the first time where we've really been able to look inside a bulk powder as it would be filled into a blister and identify where those very fine particles are located within that blend. And that's important to us because it builds up an image of the porosity of that blend to airflow. It allows us to start looking at where the high surface energy particles are distributed, and it allows us to start building up a model of how those particles are going to behave during blending and during aerosolization processes. So we're able to pull out multiple statistics with here. We're able to identify, how many fine particles are present within different types of lactose. We would be able to compare between different batches to see whether we have the same proportion of fine particles per volume. We'd be able to do things like seeing whether the distribution of those fines has changed from batch to batch. And that will allow us for the first time to start looking at performance issues and variability issues between products. And we can see some of the types of work that we can do here. So this shows you a reconstructing approach of agglomerating powders. We've taken the lactose as received. We've undertaken a high shear blending approach on that lactose without any drug, and we've undertaken a high shear blending approach of the lactose with drug. So this green line here is as received. We can see that by undertaking high shear blending on the lactose without any drug included in the system, we've ended up actually creating some intrinsic fines within the blend that weren't there to start with, so the high shear energy has done that. However, when we formulate with drug, we can see that that creation of fines doesn't occur. So what we're seeing from this three-dimensional reconstruction and pulling out the particle size distributions is that the fine particle drug seems to be protecting the lactose, the bulk lactose, from the fracture that happens during the high shear blending. Of course, this is one experiment. We are intending to look at this much farther, but with the labs shut down at the moment, we're not able to get back into the labs to study this phenomenon an awful lot more. This is something that has been highlighted previously, which is that the energy can be going into the micronized drug agglomerates and breaking those up rather than subjecting the large carrier lactose crystals to that energy. So when we start to think about what the microstructure is in the formulation, we can see we're able to now build up three-dimensional images. We can look to see the particle size distributions. We can see things like porosity of powder, density of the powder bed. We can see location of the drug distribution throughout the blend and start to really investigate what that microstructure of a powder would be and build that into our performance models, whether it's aerosolization or blending performance, content uniformity performance, to really start to inform ourselves how those materials are going to behave throughout the formulation process. So I'd like to thank all of the partners that have worked on this. Joanna at Hertfordshire and some of the PhD students who've been working on this. Our colleagues up in Manchester, Parmesh, Phil and Tim. Also at Cambridge, Chijong and James, who we heard from earlier. We've got a big team at Leeds, Ian, Vivian, Kevin and Robert, and also Digby, who started off the work with me up at when he was at Cambridge, but has since moved over and we finished the work when he moved to Canterbury. And certainly like to thank all of our industrial sponsors who've been working with us in this project, particularly the great work from Zeiss, who've made some microscopes available to us in China, which happened to reopen in I think it was about April. While we were all shutting down our labs, they were able to reopen and test some of those time-critical samples for us when we couldn't get any of the data out of the labs. And particularly, we also obviously like to thank EPSOC for funding this work. And I guess I'll move on to the questions.
Thank you, Darragh. Yeah, so we've got, so in the interest of time, I'm just going to take one of the questions, if that's okay.
Yeah, and then I can carry on giving answers through the chat afterwards.
Through the chat, and obviously your team is online as well, so they can answer as appropriate as necessary. So exactly. Just quickly, I'll just take the first one, which asked about sort of how complex is identifying particles at XCT, and has the process been automated? So
yes, I mean it is a complex process. I think what's important to say is that it's the real complexity which none of the systems to date have managed to achieve is to identify second species within the blend. How do we identify different chemical species within the blend? We have worked, and Parmesh particularly has worked with Zeiss quite closely on this. It is proprietary technology, so I can't really talk very much yet about the way that that's worked, but it's using X-ray physics to identify differences between the materials. So we can now do that. The scale of I think there was a question around the scale at which we can pick out those differences. well, we're able to identify individual particles down to about 2 microns within the microscale CT, and we can identify down to 200 nanometers within the nanoscale CT. So we do get an ability to study that right between the scale ranges, which are important for inhalation, and we're able to use X-ray physics to identify different chemical species within the blend.
Thank you very much. Okay, so if I could just ask you to respond to the other questions, that would be perfect.
Yeah, we'll go through and we'll do that through the chat for everybody as we go.
Brilliant. Thanks so much for your talk.
Okay, I'll stop sharing now and hand it back to you.